It is my pleasure to introduce all of you today to Elisa Navarro. Elisa got a degree in biochemistry from the Universidad Complutense here in Madrid, and then got her PhD at the Universidad Autónoma, also in Madrid, where she focused on the role of alpha-7 nicotinic receptors in controlling neuroinflammation. Afterwards, she moved to Mount Sinai in New York to study the role of peripheral immune system in the development of Parkinson's and Alzheimer's disease. Around 2 years ago, she came back to Spain and is currently an assistant professor in the Faculty of Medicine at the Universidad Complutense. Her talk today is titled Role of LRRK2 in the innate immune system and its contribution to Parkinson's disease. So Elisa, thank you very much for your willingness to share with us your work. Welcome to this webinar series. The floor is yours, so please share your screen and go ahead with your presentation. Thank you.
Okay, thank you very much, Miguel. I guess you can see my screen now, right?
Yes.
The presentation, perfectly. So good afternoon, everyone. Thank you very much for the introduction. So as Miguel has said, the presentation, the data that I'm going to show you today is focusing, I'm going to put 1 second, is focusing on this mutation, the G2019S mutation, which is located in the LRRK2 gene, which increases the risk to suffer Parkinson's disease. And we are going to focus specifically on how this mutation could affect myeloid function. This project is ongoing project. Most of, or a lot of the data that I'm going to show you today are preliminary, or we are still working on it. It was one of the last projects in which I was involved during my postdoc in Mount Sinai, and it's also what I would like to be the starting point here in my research in Spain. So any comments and any feedback would be more than welcome about this data. So as I have mentioned, this gene is associated to a higher risk to develop Parkinson's disease. Parkinson's disease doesn't need a lot of introduction in this audience. It's the second most common neurodegenerative disorder, and it affects around 1 to 2% of people above 65 years old, with this percentage rapidly increasing with age. It is pathologically caused by the accumulation of alpha-synuclein in a region of the brain called substantia nigra pars compacta, where it causes the loss of dopaminergic neurons. And this loss of dopaminergic neurons leads to the most common or typical symptoms of the disease, which are motor symptoms such as tremor, bradykinesia, rigidity, or postural imbalance. We also know that besides these motor symptoms, there are also a lot of non-motor symptoms. So Parkinson's disease is in the end a multifactorial disease with symptoms such as depression, constipation, loss of smell, etc. And these non-motor symptoms seem to precede to the motor symptoms and to be happening long before diagnosis of the disease. So in the recent years, our understanding of the genetics of Parkinson's disease has evolved tremendously. Thanks to the development of genome-wide association studies, or GWAS. The most recent GWAS was published in 2019 by NALS and collaborators, and the results they obtained are summarized in this Manhattan plot, where you can see in the x-axis the chromosome position and in the y-axis the p-value. And each of the dots represents specific genetic variants. In red are highlighted those variants which are significantly associated to a higher risk of developing Parkinson's disease. So with this paper, what they showed is that there are over 60 loci significantly associated to Parkinson, showing or demonstrating that Parkinson's disease has an important heritable component, which is estimated currently to be around 30%. in a more simplistic way of seeing the genetic component or the genetic landscape of Parkinson's disease is summarized in this illustration, where you can see in the x-axis the frequency of a variant in the population, and in the y-axis the penetrance of that variant into the development of Parkinson's disease. So you can see in the left part of the graph variants which are really, really rare in the population, but they have a lot of penetrance into the disease. And this accounts for the familial forms of Parkinson, which are minoritary, as all of you may know, around 5 to 10% of all cases. We are more interested in the sporadic forms, which would fall in this part of the graph, where we see that there are also associated variants. In this case, there are common variants. Some of them are as frequent as even 40% of the population carrying some of them. So this means that in this audience, a lot of us will be carrying at least some of these variants. But good luck is that the penetrance they have into the disease is really low. So the hypothesis now is that there is kind of a cumulative effect among these variants and also other environmental risk factors which predispose the person to develop Parkinson. And if you focus on the middle part of the graph, you can see here the LAR2 gene with the G2019S mutation, which has an intermediate frequency in the population, and it has a considerable risk into developing Parkinson's disease. So this variant, the G2019S, is the most common variant among sporadic cases to develop the disease. So who is LAR2? LAR2 is the acronym of leucine-rich repeat kinase 2, and it is also known as dardarin. And this name comes from the Basque word dardara, which means tremor. And it was because in the discovery of this gene, it was pivotal to genotype a lot of families in the Basque country, which were carriers of a mutation in this gene. And the name, or thanking these families, this name was accounted. So LAR2 or dardarin, it is a huge gene which encodes for a huge protein. Kind of unique, it has a lot of different domains, at least 4 domains which are related to protein-protein interaction. And then apart from these protein-protein interaction domains, it has 2 independent catalytic domains, a rock core domain here, which has a GTPase activity, and a kinase domain here. A lot of variants or SNPs have been identified in these genes. Some or a lot of them are not pathogenic, but at least 6 of them are pathogenic in the context of Parkinson's disease. So they increase the risk of suffering Parkinson's disease, and most of them are located in the catalytic domains. It must be noted, and I will come back to this later, that there are also variants which increase the risk to suffer autoimmune disorders such as Crohn's disease. So this points to a link between LAR2 and the immune system. So the variant in which I'm interested, as I mentioned, is the G2019S, which is a substitution of a glycine to a serine in this position, in this amino acid, that, as you can see, falls into the kinase domain. So it is well established now that this mutation increases the kinase activity in the patients that are carrying this mutation. And the percentage or the frequency of this mutation, it varies a lot among the different population. It is highly prevalent in Ashkenazi Jews, for example, or in North Arabs in Africa. So since the discovery of LAR2 and the implication of LAR2 in the etiology of Parkinson's disease, a lot of work has gone into understanding its functionality. So we know now that LAR2 can bind to different membranes inside the cell, such as the endothelial reticulum, the lysosomes, the endosomes. And in this membrane, it seems to be activating different downstream effectors. Among them, it must be noted the RAP-GTPases. A lot of functions has been described for LAR2, which makes its biology really complex. It is known to be related to the endolysosomal pathway, also to be implicated in autophagy, in mitochondrial metabolism, ribosome, etc. But it must be noted that most of the studies have been done in neuronal cultures, and it's not since recently that people are starting to focus on other cell types. We really believe that it's important to look to the LAR2 gene and the LAR2 activity within the immune cells. And this is because of a number of reasons. First of all, because LAR2 is highly expressed in the immune cells. It is highly expressed in lungs, in central nervous system, and also in immune cells, both in periphery and in the central immune system, in the CNS. Also, it must be noted that, as I already mentioned, variants in the LAR2 gene not only increase the risk to suffer Parkinson, but also to suffer other autoimmune disorders. I already mentioned Crohn's disease, but it also increases the risk to suffer an infection from Mycobacterium tuberculosis or to suffer leprosy. More importantly, through genetic studies, through the study of expression quantitative trait loci, it has been demonstrated in several papers that variants in the LAR2 gene, in this case are common variants, have an EQTL effect in cells of the immune system, such as monocytes or macrophages, where the risk allele does increase the levels of expression of this gene. And this is not happening in other cell types, such as the T cells, which are part of the adaptive immune system, or even in brain homogenate, giving a causality to the immune system and the role of LAR2. And finally, if we challenge cells with pro-inflammatory stimuli, although there is a lot of discrepancy in the literature about this, but the activity of LAR2 seems to be affected, although the directionality and the context still is under debate. So with all this, what we wanted to see in this project is to understand better how this mutation is affecting myeloid function. And for doing so, we are taking two independent approximations. First of all, what we are going to use is induced microglial cells derived from iPSCs, which have been edited for the G2019S mutation. Yeah, and then at the end of the talk, I will show you some preliminary data using patient-derived monocytes. So I'm going to start with this first approach. And for this approximation, what we did, we were lucky enough to get cells from a control line of pluripotent stem cells, which were CRISPR edited for the G2019S mutation. This editing was done by Alexandra Beilina at the Mark Coxon lab, and they shared those lines with us. So with these pluripotent stem cells, we decided to differentiate them to IMGLs, to microglial cells. We used the protocol that has already published by the Blurton-Jones lab, that were the first to describe a protocol for differentiation iPSCs to IMGLs. And for doing all these differentiations, this person, who was a grad student at Alison Goetz lab there at Mount Sinai, she was pivotal because she did a scientific stay in the Blurton-Jones lab, and she optimized all this protocol in the lab with us. So for the differentiation to IMGLs, we need first to get into a step of generating hematopoietic progenitor cells. Remember that microglia don't derive from the same progenitors, such as neurons or astrocytes or oligodendrocytes. So we need to get to these HPCs first, and from here derive to IMGLs in a protocol which takes around 30 days. So this is how the cells look after differentiation. It is important to note that they do express microglial markers such as P2RY12 or CX3CR1. They also express IBI1, and they express LAR2, which is really important for this project. So once we had optimized the protocol of differentiation, the first thing we did was to assess if the cells could respond to immune challenges as a proof of concept. We used LPS that all of you, I'm sure, are familiar with it at a concentration of 10 nanograms per ml. And we also use interferon gamma as interferons, both type 1 and type 2 are being related to neurodegeneration. So we thought it could be a more physiological stimuli. We stimulated the cells for 24 hours, and afterwards we assessed gene expression using RNA sequencing. So when we stimulated the cells, either with interferon gamma or LPS, we observed a huge transcriptomic response with both stimuli. Here, I'm just showing you some heat maps filtering for genes which are related either to LRH2 activity, such as the RAFs or other downstream effectors coming from the literature. And you can see how after interferon gamma or LPS, the expression of these genes changes radically in comparison to basal samples. Something similar happens if we filter for genes which have been prioritized through GWAS analysis to be related to Parkinson's disease. So these results just confirm that cells are responding to these pro-inflammatory stimuli, and moreover, that these stimuli seem to produce a relevant response in the context of Parkinson's disease. So it is a good experimental paradigm for us to perform our study. So once we have established the conditions for the experiment, we are going to focus on the effect of G2019S mutation in the LRH2 gene in terms of myeloid or of microglial response. So for that, we are going to use 3 lines. As I was mentioning before, the wild types, which are not carriers of the G2019S mutation. And then we are going to have 2 isogenic cell lines, one that has one allele edited for the G2019S mutation, that I will call them G2019S heads. And then the homes, both of the alleles are edited for the G2019S mutation. And we are going to see how these cells, which have been gene edited for this mutation, respond or differ from the wild type cells, not only in baseline condition, but also under the 2 pro-inflammatory stimuli that I already introduced, interferon gamma and LPS. So the results that we obtained, I'm going to show you in this slide. In the upper part of the slide, I'm going to show you the heterozygous cells in comparison to the wild types. And in the bottom part of the slide, I will show you the homozygous in comparison to the wild types. So the results are going to be shown in these volcano plots. For those who are not familiar with these plots, in the x-axis, I'm going to be showing the fold change, and in the y-axis, the p-value. And each of these dots is going to represent a specific gene. So anything above 0 is going to be upregulated genes in the G2019S cells, and anything below 0, downregulated genes. So when we do the comparison in baseline situation, as you can see, all the dots appear in gray, so meaning they don't meet the threshold of 0.05 significance. So we don't see any gene to be significantly dysregulated in cells which carry the G2019S mutation. So somehow this mutation is not doing anything in baseline. Nevertheless, when we stimulate the cells with interferon gamma, we start to see a lot of differentially expressed genes, around 30 in the case of the heads, but up to 500 differentially expressed genes in case of the homes. With in color red, I'm showing the upregulated genes, and in blue, the downregulated genes. Something similar, but with a more profound dysregulation, is shown or is obtained under LPS stimulation, where you can see up to 2,000 differentially expressed genes in the homes cells. It must be noted that the genes that we obtain in the heads and the homes are over 90% the same genes, so it is shared, meaning that the difference obtained here between heads and homes is kind of a difference in terms of dosage, of genetic dosage. So on, I'm gonna focus on the results obtained with the homozygous cell line. So another way of visually representing these results is presented in this graph, where you can see, as I have already mentioned, how in baseline the G2019S mutation is not producing any transcriptional dysregulation. But when we stimulate the cells, either with interferon gamma or LPS, we start to see a lot of differentially expressed genes. So these results highlight that the G2019S mutation can be silent in baseline, but primed under stimulation. And this finding points to a link between genetics and environment into the development of the pathology. So moving forward, the following question would be to understand which is the biological significance of all these gene dysregulation. And for answering this question, we used a pathway tool called GSCA. There are multiple pathway tools that can be used. We normally use GSCA. And we split the genes in upregulated versus downregulated. So I'm going to start showing you the results that we obtained with the genes which were upregulated in the G2019S cells. So when we did this analysis, we obtained a lot of enriched pathways, but the ones that were popping up more frequently and especially more significantly were the ones that I'm showing you in this plot. The most significant pathway that we observed to be enriched in the G2019S mutation is phagosome maturation, especially in the LPS condition, with other GO terms related, such as phagolysosome, lysosomal transport, protein targeting the lysosome, etc. So it seems that these upregulated genes are enriched for genes related to the phagolysosomal pathway. The other pathway that popped up really significant is related to lipid metabolism, with GO terms such as lipid transport, storage, lipid localization, etc. Again, this upregulated gene seems to be enriched for lipid metabolism genes. In an attempt of trying to do a functional validation of these results, we tried to do a phagocytosis experiment. So what we did is the same protocol that we have been using so far of IMGS differentiation and afterwards stimulation with the LPS or interferon gamma for 24 hours. And in this case, after this stimulation, we would culture the cells with some debris that we could assess the capacity of the cells to phagocytose these debris. We started using human myelin isolated from postmortem human tissues because human myelin is really rich in lipids. So we thought it could be interesting or relevant and physiological debris to assess phagocytosis. This human myelin, we labeled it with pH Frodo, which is a fluorescent dye, whose fluorescence is going to depend on the pH. So in acidic pH, for example, that happens in the lysosome, the fluorescence is going to be higher. So for example, if the cells are able to uptake the myelin and that myelin goes to the lysosome, is where we would obtain a higher fluorescent intensity. We treat the cells for 24 hours with this human myelin, and then we assess the fluorescent intensity in an attempt to measure the phagocytosis capacity of the cells. So here in this video, I'm gonna show you, you can see the cells, how they look. They are super cool because they are constantly moving around the plate. And then at some point we add the myelin that you see is red, and you see how the cells kind of change the morphology and they move towards this debris. So 24 hours later, in the presence of this myelin, we assess or we measure the fluorescence, which could be which we believe is a measure of phagocytosis. And the results that we obtained are in line with the results that I showed you at the transcriptome level. We see how the wild type cells that would be here in pink do uptake or do phagocytose myelin, but they do it in a lower rate than heads or horns. that you can see here, how in the 3 different conditions, not only under stimulation, but also in baseline, heads and homes do have a higher fluorescence rate, meaning that they are uptaking more myelin towards the lysosome. The process of phagocytosis is complex. It needs, when the debris needs to be uptaken, and then it needs to be metabolized, and then it needs to be degraded. So we don't really know where these differences are coming from, or if it is in all the process. But in trying to answer this point, we decided to do the same exact experiment, but instead of using human myelin, we decided to use carboxylated beads. These beads have the key point that they cannot be degraded, they don't go to the lysosome, and they are not metabolized within the cell. So it is a measure that people or researchers use in the literature in order just to measure the uptake capacity. So we did the same exact paradigm, and the results that we obtained in this case are pretty different to the ones that I have shown you in the previous slide. Here it was the same number of experiments with the same differentiation that the previous one. Here we obtain no significant results between the different cell lines. You see here the wild type, the heads and the homes. It seems to be a tendency to be the opposite. It seems like heads and homes could have, but again, it's no significant, but they have a tendency to have a lower uptake capacity, even though again, it's no significant. So that differs from the previous experiments. So we really want to dive into these differences, but so far we think that there are not going to be difference in the uptake, but the difference are going to be in the metabolism, which would be in line with also the increased lipid metabolism genes that we have seen in the transcriptomic analysis. Okay. All these genes, all these lipid metabolism genes and phagocytosis genes remind us to all these new phenotypes of the microglia that are being described in the literature. You know that microglia are really dynamic, and they are kind of changing all the time to different phenotypes depending of the context in health and disease. So the first phenotype that was described thanks to the use of single-cell technologies was defined by Karen Saul, and is the DAM microglia, the disease-associated microglia, which was in the context of Alzheimer's disease. After that, a lot of new different phenotypes are being described, like the LDAM, lipid droplet accumulating microglia. This microglia is microglia which appears during aging, and it is associated to be accumulating lipid droplets within the cytoplasm. And this microglia, when differentiated from the other, it has a total different phenotype. And finally, we also focus in another one of the phenotypes, the activated response microglia, which was described by Bart Strupper lab. It's also in the context of Alzheimer's disease, and is the microglia which is associated to the amyloid beta plaques, which again has a total different phenotype in comparison to the microglia which is not associated to these plaques. So we thought that all these genes, all the dysregulation that we were seeing was kind of similar to all these phenotypes that were being described in the literature. So we decided to do an enrichment analysis in order to see if the genes that we were getting, the differentially expressed genes that we were getting, were enriched for the genes related to all these different phenotypes. And indeed, we observed how, specifically under the LPS stimulation, the cells that express the G2019S mutation are enriched for these genes. So somehow these results are pointing to that the G2019S mutation is somehow capturing the transcriptomic alterations which are related to these microglial phenotypes associated to brain damage. Moving forward, we did the same analysis, the same approach, the same pathway analysis, but now instead of using the upregulated genes, we are gonna focus on the downregulated genes to see what is going on with them in the G2019S cells. So what we obtained, the pathways that we obtained are represented in this graph. And the significant ones were related, all of them, as you can see, to mitosis, to pathways of cell cycle, division, proliferation, or especially mitosis. In order to try to validate these transcriptomic results, we did an experiment in which we assessed the proliferation capacity of the cells using an incucyte, a microscope that is coupled to the incubator, and we assessed over time how cells were proliferating in the plate. And we observed how in pink, the wild-type cells are proliferating at a lower rate, to a higher rate than heads or homes, which have a lower rate of proliferation in comparison to the wild types. So this is somehow in line with what we were seeing at the transcriptomic level. In this line, we did also a transcription factor enrichment analysis in order to see if all these gene dysregulation was linked to a specific risk, to a specific transcription factors. And when we did this analysis, we obtained a number of transcription factors that I'm not gonna talk about. I just wanna focus your attention here. In the transcription factors, or in the family of transcription factors that seem to be more significantly enriched, controlling all the genes that are being dysregulated in our data set. Is the E2F family. You see there is significantly enriched in all the three situations, and in all the cases is downregulated in the G2019S cells. So the genes that are controlled by this transcription factor are downregulated in the G2019S cells. So E2F is a family of transcription factors. There are from 1 to 8 different members. So we assess the level of expression of this transcription factor in the IMGLs, and we observe that all these 8 members can be detected in RNA sequencing, being the one more expressed, E2F4. That, as you can see, is also the one whose the genes that are controlling are the ones that are highly expressed in the transcriptomic data. We don't see in any of the cases, as you can see in the plot, difference in expression if we compare the different genotypes. So this means that the dysregulation seems to be at the activity. So somehow the activity of E2F seems to be altered, but not at the expression level. We did also compare this expression data with published data of human microglia isolated from human samples from Gosselin. In this data set, we can also detect the 8 E2F members, but as you can see, same as we obtained, E2F seems to be the one that is highly expressed in microglia, which is cool somehow to get the same results in these IMGLs, which are kind of artificial in comparison with microglia isolated from human samples. So these results, we need to dive more into understanding better the functional consequences. But so far from this first part of the talk, what we can conclude is that G2019S mutation drives profound transcriptomic changes in human-induced microglial cells, especially under pro-inflammatory stimuli, but not in baseline. The G2019S mutation seems to be increasing the expression of genes which are related either to Phago lysosomal pathway or to lipid metabolism pathway. And the G2019S mutation, on the other hand, is decreasing the expression of genes related to mitosis or cell cycle, especially through the E2F family of transcription factors. These results open the door of new experiments, and we really believe are good, but they have limitations, especially the fact that it's only one cell line and is missing, let's say, the population or the disease, the patient's diversity. So in an attempt of trying to capture what is happening in patient-derived samples, we started the second part of the project, which is really preliminary, but I'm going to show you what we have so far, which is trying to study the effect of G2019S mutation in patient-derived monocytes. Why are we using monocytes if we are interested in microglia? So getting human microglia from patients is really difficult. There are a lot of logistic issues. You have to use postmortem samples, but there has to be a very short postmortem interval. So it is not easy, and especially if we are focusing on a very specific population of carriers of a specific mutation. So we really thought it was difficult to get enough patients, postmortem patients with the G2019S mutation, and that's why we decided to use monocytes as an approximation to microglia. They have a lot of similarities. They derive from the same progenitor. They also share similar functional and transcriptomic similarities. So we really believe they can be a good approximation to microglia. Indeed, we have shown in previous experiments or in previous papers that not only microglia, but also monocytes are enriched for the expression of EQTLs. So same experiment that I showed you at the introduction, but here not looking to just one gene, but to a lot of genes that have been prioritized in the context of Parkinson's disease. And you see how in orange microglia and in green are the monocytes, how both cell types are enriched for these EQTLs in the microglial or in the monocyte population, and not in other cell types such as here is brain homogeneity. So these results highlight that not only microglia, but also monocytes are enriched for this genetic predisposition. Also, in another published paper that we did a comparison of human microglia from postmortem tissues and monocytes from blood samples of Parkinson's disease patients and age-matched controls, we showed how microglia had a profound dysregulation in Parkinson's disease, shown in this volcano plot, which is more or less expected with all this immune implication on Parkinson's disease. But we also show how monocytes, which are these peripheral immune cells, show a profound dysregulation in Parkinson's disease. So these results show that peripheral immune system is also altered in Parkinson's disease. So these few slides, they were focused on convincing you that monocytes can be a good tool to study immune system in patient samples and much more accessible than microglia. So this is what we decided to do for this project. We decided to try to recruit Parkinson's disease samples and age-matched controls with not known neurodegenerative disorder, not known autoimmune disorder, that would be carrying or not carrying the G2019S mutation. We would get for this project between 3 or 4 tubes of blood. From this blood, we would isolate using a Perkov gradient the PBMCs, the peripheral blood mononuclear cells. And from these PBMCs, we would isolate using CD14 magnetic beads the monocyte population. After this isolation, we would do the stimulation with LPS or interferon gamma, same experimental outline as for the IMDNs. So in this slide, I'm showing you how many samples have been included so far in the results that I'm going to show you. Again, this is preliminary, it's ongoing, and I really hope to start to build a cohort here in Madrid of donors. For this part, we got a total of 9 controls and 12 Parkinson's disease with none of this group carrying the G2019S mutation. Then we got 6 non-manifesting carriers, meaning people that don't have Parkinson's disease or any other autoimmune disorder, but they do have the G2019S mutation. This population here is the hardest to find because most of people don't know, or if they don't have the disease, they don't go to the clinic. So they are real. You have to genotype a lot of samples to get those, and still we got a pretty small number. 6 is really low for population-based studies, but it's what we have so far. And then in the Parkinson's disease group carrying the G2019S mutation, we have a total of 8 donors so far. You can see that the groups are pretty balanced in terms of age. There is some disbalance. We always obtain this in the Parkinson's disease group because it's a disease that affects more males. So we have more PD males than females. And in the control, sometimes the opposite because it's the spouses of these donors. Anyways, super quickly, these are the 2 last slides. We are really limited in sample size, but so what I'm showing here is nominal p-value. But we see the same trend as we observed in the IMDS, how in the baseline situation, there is no that transcriptional dysregulation, but when we stimulate, in this case, monocytes with interferon gamma or LPS, we start to see a lot of differentially expressed genes. Same results, but in another way of visually seeing is this QQ plot where you can see how under stimulation cells separate from the expected p-value. I'm not going to focus in these plots. I'm going to just drag your attention to LAR2. We were interested in seeing if these patients have difference in expression of LAR2, but they don't. The carriers here do express the same levels as the non-carriers, no matter if they are in baseline, under interferon gamma or LPS. So we haven't seen changes in expression in comparing some of carriers and not carriers. So here is more complex to do the analysis because this is carriers or not carriers, but we do have 2 different diagnoses. So we decided to split the samples and do pathway analysis, comparing the LAR2 carriers in the control group and then the LAR2 carriers in the Parkinson's disease group. So in other ways, it's non-manifesting carriers versus the controls, and then PD LAR2 carriers versus the Parkinson's disease sporadic without the mutation. And we did pathway analysis using the same approach as before. And apart from pathways related to immune system, it was really cool to see that we obtained again, although very limited in terms of significance, remember, but pathways related to lipid metabolism and to phagolysosomal pathways. So somehow it's capturing the same dysregulation that we have seen in the IMDS, although again, these results really need to be confirmed. So from this second part, I have shown you how the G2019S mutation is also altering gene expression in patients-derived monocytes under pro-inflammatory environment. And it seems, although needs to be confirmed, how these pathways are related to lipid metabolism, phagocytosis, and immune response. And overall, the conclusion would be that G2019S mutation drives profound transcriptomic dysregulation in central and peripheral myeloid cells. And so far, apart from increasing the sample size, I'm really interested in doing more functional validation. Especially, it would be really cool to do lipidomics in order to see how is all the lipids, lipid graphs, etc. in these cells. Dive into the phagocytosis process and also perform LR2 kinase activity. And future experiments that I would love to do would be try to do pharmacological rescue, either with kinase inhibitors, which are being studied in clinical trials now, or any other pharmacological approaches. And also I would love to do some in vivo mouse model, although this kind of difficult because of the size of LAR2 and how to deliver the gene to the CNS. But all of these hopefully will be happening in the upcoming years. And with that, I would like to thank everyone who has being part of this project, both in New York, where all these experiment started, and now at Javier Fernandez Reed Lab, where I plan to continue with all this story. And with that, thank you very much for your attention, and I will be very happy to take any questions.
Okay, thank you very much, Elisa, for a very exciting presentation. So the paper is now open for discussion. As I said earlier, if anyone is interested in making questions to Elisa, please do so by either raising your hand using the application or by sending me a message through the chat and stating your interest in making that question. So while we wait for the first question, let me remind everyone that CIBERNED Webinar Series are held every other week on Thursdays at 3:30 p.m. And if you're interested in checking upcoming webinars, you can do so by going to the CIBERNED website, and you'll find the program for the next few weeks. I believe the program is ready until the end of the year. Also, the recordings of this seminar will be posted in the CIBERNED website. So just in case anyone missed part of it, you can go there and double check that in a few days. Okay, so we have first question here from Nacho Torres. I'm not sure whether you have a microphone, Ignacio, so you can do the question yourself, or you want me to read it?
Whatever, I can do it.
Okay, go ahead, please.
So you tested two specific stimuli which are pro-inflammatory. Did you use another type of stimuli just to be sure how specific it works? Maybe any type of stimuli will produce changes under the mutation.
Yeah, that's totally possible. We haven't tested any other stimuli. We just used the two pro-inflammatory because of this link that we believe is important between immune system and the LAR2 gene. We have thought of using other stimuli, but linked to Parkinson disease, like alpha-synuclein fibrils or things like that. But we didn't think about seeing the specificity, which is a good point, using any other stimuli and see if we do have that dysregulation or not. It's a good point.
And a second question along the same line is that you have seen similar changes in monocytes and microglia, but how about all other cell types? Because maybe these type of changes are also seen in other cell types. How specific, again, are for these cells?
No idea. You mean neurons, for example, or astrocytes?
Yeah, whatever, or even peripheral cells.
Or peripheral. We haven't done, we have there in New York another project that they are going to look at T cells. So again, immune system, but adaptive immune system. But I have not an answer for that because we haven't tested. LAR2 plays a role in other cell types. It has been studied more in the central nervous system because of the link to Parkinson disease. So it's been studied in neurons, in astrocytes, and but how they respond to stimuli, I have not an answer about that. Again, because we were looking for this immune regulation or the role that LAR2 could have in the immune regulation. But it's a very good point for sure.
Okay, thank you.
All right, thank you both. I think we have another raise hands here. Let me see. Yeah, Teresa Iglesias. Teresa, you want to go ahead and make a question?
Hello, Elisa. Thank you very much for your talk. That's really nice. A lot of work. I'm just curious to know, did you expect not to find any changes on the basal having the mutation? Because knowing that it's an active kinase with a mutation, I would expect to find some changes in the transcriptional profile of the cells, right? So
yeah.
I wonder how were your feelings when you found that basal. So it makes you think that maybe this activity is not making any difference or having any role, at least at basal conditions, and then you need to have an inducer to have this kinase activity making a damage to the cell? Or what is your thoughts, or are your thoughts on that?
Yeah, we knew, we kind of were looking for that. I mean, we didn't know what was gonna happen. What we knew before doing the experiment is from the other project that it was monocytes, it wasn't microglia, that we had a lot of patients with Parkinson's disease, and some of them were carriers of the LAR2 mutation. And when we did the filtered those cells and we did the transcriptomic analysis in this other approach, we didn't get to see anything, any differentially expressed genes. That's why we thought, how is not happening anything if this disease, or if this mutation is supposed to be increasing the risk of the disease? And that's why we decided to stimulate the cell. So somehow we knew that LAR2 was important for the immune system. So that's why we decided to challenge the cells. But we didn't expect to have this like profound difference. And that was kind of surprising, and I really like, because it really links the genetics and the environmental factors, and it's something that we hear a lot in the clinic, right? That people that you can live with a mutation with nothing, and then when you suffer from anything, you go to the hospital, wherever, some challenge, and they start to develop the disease. So I see something like that in this context, how this suffering I don't know, an infection or something different, not linked to the disease, but how this gives you the predisposition then to start developing Parkinson. So I don't know if I have answered. Like, somehow we were looking for that challenge in the cells. We thought that we could like stimulate the differences, but we didn't expect to have this profound difference.
No, it took my attention, just thinking that if you have, kinase that is not supposed to be active, or otherwise maybe you have it active anyway in basal conditions, and then
it is supposed, yeah, in basal conditions, the kinase activity of this protein is higher. That has been demonstrated. I want to measure in the monocytes. We haven't done yet, if that's the case, but it's what is reported in the literature, that people carrying the mutation have a higher activity in baseline. You don't need to stimulate it.
Yeah, I mean.
So I guess it's the role that LAR2 is playing, in baseline. Maybe it doesn't have that important role, but it's when you stimulate the cells, when they need to respond to this immune challenge, when LAR2 plays an important role, and it's when you start to see the difference in the mutated cells. That would be the hypothesis that I would give to this.
I know I have to give a thought.
Any thoughts that you have, I would be happy to hear in the future.
Yes, welcome.
All right. Okay. Thanks, Teresa and Elisa. So any more questions? I don't see any raised hands or any message coming through the chat. Yeah, now Nacho Torres again.
Sorry for asking so much.
No.
I have another question. If nobody else is questioning, so I was surprised also to see that the inflammatory stimuli didn't activate the phagocytosis in the wild type microglia. Wouldn't you expect this stimuli to stimulate also phagocytosis?
What do you mean?
Using the myelin, the human myelin, to do the phagocytosis, and in the wild type microglia, you didn't see any stimulation with LPS or interferon. If I remember well the graph.
You mean that we didn't see differences between the LPS or interferon and the wild types in terms of phagocytosis?
Right.
Yeah, I didn't expect that. And it has been reported in the literature the contrary, that under pro-inflammatory stimuli, cells have a higher capacity of phagocytosis.
Right.
So I didn't expect this. And I don't know the reason.
Yeah, I would be expecting something like that. Yes, I'm surprised too. Yeah.
Yeah.
Thank you anyway.
I mean, also, the phagocytosis is an N of 5, so it has been repeated 5 different differentiations, but still a lot of this is preliminary, like it's only one cell line, for example, that has to be only coming from one donor. So again, we are gonna build from here, so take it cautiously also.
Okay, thank you.
All right, thank you, Nacho. Any more questions? Yeah, again, I don't see any raised hands. No message through the chat. So that would be your last chance to make a questions to Elisa for today. So no more questions. Okay, so if there are no more questions, I think it's time to close the session, but not before thanking again Elisa for a very nice presentation, for her willingness to share with us her results, even the preliminary ones. Very interesting. Thank you everyone for attending, and I wish you all a very nice rest of the afternoon. So I'll see you next time.
Thank you.
Bye-bye.
Bye.